talk about more detail in Prophase 1. Why it is necessary? Because Prophase 1 is the longest, most complex stage of meiosis. At this stage, homologous chromosomes pair up, exchange the genetic material, and condense together, which is essential for genetic variation and proper chromosome separation. Now we'll talk about what already has happened. After interphase, okay, we know that DNA is already replicated, which means like this. Okay, each chromosome consists of two chromatids. Chromatid 1, chromatid 2. These sister chromatids are held by together by the cohesion proteins. These sister chromatids held together by cohesion proteins. So let's discuss more about crossing over and the synapses in Prophase 1. Suppose suppose this is chromosome 1. Okay, this is from the paternal side, we can say father side. Okay, and and this is from the mother side, we can say maternal. Mother side. And let me just term other things also. These things are getting hold by, okay, the chromosome get hold by some things that as cohesions. Okay, this long gap is known as centromere. At this point, the two chromosomes are joined together. We can say at this point, the chromosome, we can say sister chromatids are joined together. Okay, they are both called as homologous pair of homologous chromosomes or homologs. I think you can call them. And now what happened is, at some point here, for example, like at this side or this side, what happened is, DNA gets broken down. You can see this side, at this side and this side, the DNA gets broke. DNA breaks down. Now what happened is, now you can see more clearly. Now a different type of thing happens here between these two chromosomes. At this point of stage, you see that DNA breaks here. Now they come close together, and when they come close together, a zipper like protein complex, what we called it as zipper like protein complex form here. Okay, and this thing we can call it as synaptonemal complex. Synaptonemal complex here. Okay, what this do, like these are things trying to bring the chromosomes together. Okay, and the sister chromatid together. What will happen next is, the red part that has been cut from here, and here also, this will attach here like this, and from this red part will attach here. And from the opposite side also, the blue part is will attach here and here also, and then this thing is referred as crossovers, or in the or we can say crossing over occurred. Crossovers. Okay, what was mainly happened is, see, suppose this is father side, this is mother. Okay, now some of the part of this side, we can say mother side, get cut. First of all, let me just term it as non-sister chromatid. This is chromatid number 1 from the father side. This is chromatid number 2, which is I call it as 1 dash. This is chromatid number 2 from the mother side. This is chromatid number 2 dash. These 1 dash and 2 are called as non-sister chromatids, because here they are not from the same family. Okay, they are not from the same chromosome, they are from different chromosome. The bond between them, it is called as synapses. Now they are forming a bond. How? As this part of chromosomal material, we can say chromatid, gets attached to 2, and 2 part gets attached to 1. Okay, like this. Now you can view it clearly. At this part, synapses happened. Synapses happened here. Plus, after these, now there are 4 sister chromatids. We will call them as tetras and bivalent. Chiasmata also, as this point where the 2 chromosomes are attached together by sister chromatids, known as chiasmata. In one chromosome, you may found like 1 or 2 chiasmata junctions. Now, basically, what happened later? Now you can see the junctions more clearly. First junction, second junction, both called as chiasmata. At this point, the crossing over occurred. Okay, and then what happened? The chromosome continued to condense and move toward the metaphasic plate. After this, now we'll discuss a key comparison between the mitosis and meiosis. Now, suppose these are the parent cell. Okay, before duplication. Before chromosomal duplication. We refer them as diploid. I have written 6 here because there are 6 chromosome present inside the cell. Okay. Now after this stage, both in mitosis and meiosis, what happened is at this point of stage, the chromosome get duplicated. Duplicate. You can see the both duplication has been differently occurred here. Now, what just happened here is, see, here a duplication occurs and duplicated chromosomes have two sister chromatids. Okay, same as occur here also, but here the thing is different. The site here, we can see the chiasma here, chiasma, site of crossing over, and also the pair of homologs held together by chiasma and sister chromatid chromosome. We can see the pair of homologs held together, and but we mainly see crossing over occurred. Here no crossing over occurred. Now next come to next stage. This was the prophase. Let me mention here prophase and prophase here also. Now what happened in the metaphase? What you can see in the metaphase and metaphase 1 is, see, in metaphase, the individual chromosome get lined up at the metaphasic plate. See, individual chromosomes here. However, in the metaphase 1, and like you can see in the meiosis, the pair of homologous chromosomes line up at metaphase plate. Here the pairs get lined up. Metaphase plate. Okay. Now we'll discuss about anaphase and telophase. At anaphase and telophase in the mitosis area, what happened in the anaphase is, we can see here sister chromatid gets separated from the mitosis. In mitosis, the sister chromatid gets in half from the centromeric cohesion. Cohesion breaks here. They can separate it into two different cells after the late telophase to and beginning of the cytokinesis, and sister chromatid separate. Thus, we form two daughter cells consist where they are diploid daughter cells in mitosis. However, the case is different in the meiosis part. Now, when you see here is the homolog gets separated in the anaphase 1, and sister chromatid remain attached in the centromere. Sister chromatid still attached at centromere. This is centromere. They're still attached here, but the homolog gets separated here, so they become haploid now. N is equal to 3. And towards the end of the telophase, they divide further again, and what happened is we can see like the two daughter cells of meiosis 1 has formed here. In the meiosis 2, now at the end of the meiosis 2, we can see that like sister chromatid gets separated also after the anaphase 2, anaphase 2 and meiosis 2 end. Sister gametes get separated, and then each one of form is haploid. Thus, four cells, four daughter cells get formed here. Four daughter cells. Sister gametes separate. Okay, after N of phase 2, and this marks as the end of complete meiosis. Now let's talk about the basic difference. See, so what happened is the type of cells in the mitosis mainly are body cells. We can say somatic cells which undergo cell division, and then meiosis there are germ cells. Mainly we can call them as gametes. Okay, the chromosome sets it is nearly conserved. Okay, and diploid to diploid again. In the meiosis it is reduced diploid to haploid. The number of diffusion take place in mitosis is 2. Sorry, then the mitosis it is 1, and the meiosis it is 2, where at meiosis 1 where daughter cells get divided into 2, and in the meiosis 2 the daughter cells get divided into 4. Okay, daughter cells are 2 here. In meiosis the 4 daughter cells form genetically identical, nearly same. Identical means nearly same or nearly exact copy of the parent. It is genetically different from the parent. Okay, this is the key idea to know this in mind. Mitosis generally what do it preserves the chromosome number. The chromosome number doesn't change from diploid to haploid or haploid to diploid, but in meiosis chromosome number get reduces, which leads to variation. Now let's talk about the role of sister chromatid cohesion. In mitosis, like the cohesion holds sister chromatid together. At the end of the metaphase, enzyme cut the cohesions. The enzyme was separase, and the sister chromatid separates in the anaphase. However, in the meiosis, cohesion is done in two steps. First, in the anaphase 1, where cohesion along the chromatid arms get released and the homologous chromosome gets separated, sister chromatids stay together. Then in the anaphase 2, cohesion at the centromere get released and then the sister chromatids separated. Okay. Now we'll talk about the chiasmata. Little bit information about this. In chiasmata, why the homologs stay together? This is because during the crossing over plus the cohesion that create the chiasmata, and the chiasmata, what does this do? This helps to hold the chromosomes which are homologous in nature together and allow the correct alignment at the metaphase 1. Without the chiasmata, no correct alignment is done, which leads to improper separation. We can say non-disjunction. So that's why it is necessary. Okay. So the next lecture will be the final lecture for the meiosis part, and then we'll tune to Mendel area, Mendel and the gene idea. We'll discuss about the Mendel achievements and how he held the genes, and about discuss about the pea plants. That's it for today. Hope you guys enjoy it. Have a good day.